Okay.
All right.
Yeah. Okay, thank you very much for giving me this opportunity to present my work here. So today I'll walk you through the, our in vivo RNA Structurome project. So, RNA structure plays a very important role in biology, and it has, very important role in the regulation role in transcription, translation, and RNA degradation. Traditionally, we probe RNA structure one by one in vitro in gels, and now we want to probe a lot of RNAs at a time, and in living cells, which may include a lot of RNA-protein interaction. So this is our pipeline. So first, we need to build a Arabidopsis seedling and extract RNAs and apply the DMS and dimethyl sulfate, which can methylate A's and C's when they are located on the single-stranded part of the A's and the C's. And after that, we do reverse transcription, which will stop at this methylated site. Then we do ligation and PCR and put it on a linear sequencing machine. So after that, I developed this computational pipeline. So first, we map all the reads reference genome, and then we calculate the number of the RT stops that target each nucleotide. By targeting, I mean that's the one nucleotide going before the mapping position of this RT stop reads, which is a real methylated site. So after this, we do data normalization and develop the statistical model to derive the reactivity. And using this reactivity, we predict RNA structures, and also we can predict RNA structure directly from RNA sequence. So that computational pipeline is integrated into a StructureFold platform, which I developed, which is now a part of PSU Galaxy system. So if you have your own RNA structure profiling data, you can put it on this platform, and it can do bioinformatics analysis automatically. So we really provide a very large set of RNA structure information at nucleotide resolution. So for each color dot here represents the structural information on one nucleotide on one RNA. So the higher reactivity of a nucleotide, it shows that more likely will be located on a single-stranded part. So this is a part of the 18S ribosomal RNA phylogenetic structure in Arabidopsis, and we can see most of the red nucleotides, which has the highest reactivity, are mainly located on single-stranded part. We also find a lot of interesting biological findings. The first is a very interesting triplet pattern of DMS reactivity throughout the CDS region in Arabidopsis, which is confirmed by the Fourier transform. And the second, we see an elevated structural level going just before the alternative polyadenylation site and a decreased RNA structural level around this site. We have also observed an increased RNA structural level just at a 5' splice site only for these RNAs that are not spliced at the site. And for these RNAs that flow differently between in silico and in vivo means we use restraints reactivity to fold, and in silico we just use a sequence to fold. If they're different, we find they're more related to response to stress, but if they're similar, they're more close to housekeeping genes. And we know RNA has structure, but that's made of nucleotide. And protein also has structure, but it's made of amino acid. These two really seems to be irrelevant. But interestingly, I do find that there's some relationship between these two structures. And for these regions that code for protein domain junctions has lower reactivity as compared to these coding for domains. That shows elevated structural level at this site. And because of that, that may slow down the translation rate and give enough time for the already encoded protein domain to fold into the correct form. And also because of that, that will cause clustering of ribosomes at this site that may provide protection on this site, so may prevent the DMS from targeting this site. So we also find a decreased DMS reactivity pattern at this region. Okay, to sum up, so we have developed Structure-seq, which can probe a lot of RNAs at a time in living cells, and we also developed, a Structure-seq, a StructureFold, which can, do bioinformatics analysis, and we find a lot of interesting biological stories. So I want to have my great thanks to my co-workers, advisors, and also all my lab mates and my findings. Thank you for your attention.